And now we're going to take a closer look at the actual structure of the complex that binds antigen and allows a T cell to recognize it. If you'll recall from the last lectures we did on the T cell receptor, it does not undergo affinity maturation the way an immunoglobulin receptor does. And for that reason, in general, it doesn't have as tight a binding property as does a well-developed, improved immunoglobulin receptor. So that when we take a look at this T cell receptor, we're going to look at some, as it were, assists that help it in its binding. If you'll recall, this recognition site up here for the alpha and beta receptor binds a peptide, and that peptide must be presented or displayed on an MHC molecule. So it's a spread out string of amino acids that either lies flat or bows out. And the recognition site has to be able to recognize a huge spectrum of different peptide sequences. And of course, one receptor will be specific for one very defined peptide sequence, and there will be lots of different receptors. Also, similar to the immunoglobulin chain, you can see that the membrane spanning regions in here do not extend very far into the cell, so that this T cell receptor also needs co-receptors to help it signal. Sometimes the entire complex is called the CD3 complex. Sometimes CD3 proteins refer to the assist co-receptors and not the TCR. In general, well, you're not going to get this thing on the surface with just the co-receptors by themselves. So here we have the overall complex. So let's take a little bit of a closer look at it. You've seen the alpha-beta receptor before. We have two immunoglobulin side chains, a membrane-spanning region, and there we have it. It's going through the plasma membrane here. Unusual in this complex is that we have charged molecules that are part of the amino acids that span this membrane, and the positive charges are indicated here, and if you will notice, the co-receptors have negative charges. So while this thing is spread out to give you a good look at it, the point is that what you're really going to have is a cluster, sort of like a bouquet of molecules associated with the membrane spanning regions of the alpha-beta receptor, so that when there are conformational changes that occur when this thing binds, there's going to be that same communication across the membrane to the cytoplasmic regions of this receptor, and you can see we have those immunotyrosine activation motifs, which is to say places where you put a phosphate on there and then begin to trigger a series of internal enzyme activations at the interior of the cell that allows for the signaling. Okay, so just for some details, we've got a complex of 3 dimers. Over here we have an epsilon delta. Note that each of these has its own immunoglobulin chain or domain on the outside of the cell. We have on the other side a gamma with that same epsilon again. So we're going to have 2 copies of the epsilon. In this part we have the gamma and the epsilon together. And again, they both have immunoglobulin domains at the exterior. Down here, however, we have what's sometimes called the zeta-zeta, or it turns out sometimes the zeta-eta. Down here, that has no immunoglobulin chain, seems to associate fairly directly with the membrane spanning regions of the receptor, and contains even more opportunities for signaling to the interior. Okay, so let's look at these two again. The zeta-zetas down here are sometimes a zeta-eta. And it turns out that the gene that codes for this protein is one of the rare ones in this whole setup that has the ability to undergo 2 different forms of splicing. So I can take the preliminary message and splice it one way and get a zeta. If I splice it in a slightly different manner, I get an eta. And so what I'll have here are two different splicing isoforms of the same gene. So again, the peptides here, the epsilon, delta, and gamma, and also the alphas and betas, are all part of the immunoglobulin superfamily. The zetas or zeta etas are not. They only have 9 amino acids to the exterior. That's just a wee little thing that comes up in here that seems to associate with part of the TCR. The negative charges on the associated peptides cause all of these things to sort of come together like a bouquet. So that while I have these things arrayed out in a linear manner so that you can get a look at what's going on, when this whole thing is together, what you're going to have is a bunch of signaling molecules that surround the alpha-beta receptor. When the alpha-beta receptor undergoes a conformational shift, all of these guys are going to start picking up phosphates and leading to some very, very interesting changes. Among them, we will see will be the relocalization of the whole receptor complex. So this is more than just turning on a switch. It's also turning on something that changes the location of the whole complex. And we'll get to see more on that later. The ITAMs function in much the same way that they do in the beta receptor. We're going to see that they attract tyrosine kinases and the autocatalysis of the transfer of phosphates, and the phosphates will then get transferred elsewhere, and that's going to lead ultimately to the activation of the whole cell. Now, the problem with this, as I mentioned, is that the recognition site does not quite have the strength to bind the peptide that the recognition site does on an immunoglobulin receptor. And because of that, we're going to have presentation on MHC molecules, and we'll also have a little assist from the co-receptors. So here are the co-receptors, the CD4 and the CD8. And both of these guys are going to go from the T cell, which will be down here. This will be the membrane of the T cell. They're going to reach around the receptor, and they're going to bind at the base of the MHC molecule. That is, these guys will be there as a kind of backup stabilization to the complex of MHC, antigen, TCR. Will be in those kind of rough locations. So let's look first at the CD4. This is used by TH cells to bind class 2 MHC. It's an immunoglobulin superfamily protein, and these lumps are each immunoglobulin domains. One of them is rather small. This one functions as a monomer, and so there's one of these peptides anchored in the membrane of the T cell. We'll see initially at a place different from the TCR. It has 4 extracellular immunoglobulin domains, and here's the N-terminal up here. That's going to actually bind to the base of the MHC2, and it's going to kind of stick into a hydrophobic pocket where the junction of the alpha-2 and beta-2 domains are. Okay, we've got a transmembrane region and a C-terminal that has 3 serines that can be phosphorylated. So let's go look at the whole thing. So here is a different picture. Here we have the T cell up at the top and the presenting cell at the bottom. And so the presenting cell here has put an antigen in bright red onto its MHC class 2. And the TCR has recognized it. So by dumb luck, this TCR has a sequence at its recognition region that binds to this antigen. And they bind. And when that happens, the TCR activates and does something that allows the CD4 to come in and stabilize the whole complex. So here's the CD4. Here's the N-terminal in the hydrophobic pocket. Here's the C terminal sticking into the interior of the TCR, and it's going to again have the ability to phosphorylate that. Here we have the MHC2 at the bottom that's anchored into the membrane of the presenting cell. And when you get these two guys together, then you go to the next step in activation, which we'll look at again later. So this is the one for the CD4, which activates the TH cell and leads to the activation of the whole and coordination of the whole immune response. In addition to that, we have the CD8. The CD8 is going to interact with MHC1. So this thing is used by TC cells to bind class 1 MHC. It is an immunoglobulin superfamily protein, but only on the N-terminal. The rest of this molecule does not have immunoglobulin domains. It functions as a dimer. You can see that it's held together by two disulfides, and it also has regions in the cytoplasm. Again, this is the T cell on this side that can be phosphorylated, and then we'll see participate in signaling. So this thing can function as either an alpha-beta heterodimer or a homodimer, linked in both cases. It sticks out from the membrane kind of like lollipops on the end of the stalk. And these two regions here together bind to the MHC1. They seem also to bind to the A3 part, but they also touch and interact with both the beta microglobulin and the A2. So let's take another closer look at that. So here is the comparable version that we had before. In this case, the TC cell is up at the top, and the presenting cell, displaying cell is down here at the bottom. In this case, you would have any one of a number of different kinds, most of the cells in your body displaying an antigen, shown here. The TCR is binding to that antigen, which implies that antigen is of some foreign origin, or there wouldn't be a TCR to recognize it. And so when the TCR recognizes this foreign antigen, here's the CD8 coming in and stabilizing this response. And you can see that it is interacting significantly with the lower part of the molecule. And the A3 is down here. There's the beta microglobulin. And this whole complex then has become stabilized. So this is how we begin, and it's certainly just the first stage of all of this. And this is the way that a TC or a TH cell recognizes antigen. The process that leads to this is supposed to get rid of anything that recognizes self-antigen in here, and it's supposed to get rid of anything that does not recognize self-MHC, the one down here. And so we will see, as with many biological generalizations, it ain't necessarily so.